To get to the NCBI Gene homepage, search for NCBI Gene and it should be your first hit. From the Gene homepage, search for BRCA1. Usually the human entry will be your first hit, but note that Gene contains entries from many organisms, so make sure you're aware of that. Clicking on the BRCA1 link will take you to the report page for that particular gene. Above the summary table, the gene ID, which is a number, is displayed. The gene ID for BRCA1 is 672. This provides a unique ID for the gene in the database. Let's look at some key attributes of the gene in the summary table. Its official symbol provided by HGNC is BRCA1, and its official full name is listed as well. The gene type will tell you that this is a protein coding gene, and the RefSeq status will give you an idea of the confidence in the gene. Reviewed means good confidence. The also known as information will tell you other names this gene is or has been known by. Searching one of these terms should probably return this gene as well, although probably lower in the list than if you were to search by its official symbol. In this case, you see iris, which is there's a fruit fly gene known as iris. It's the first hit, but our human entry BRCA1 is the second entry here. So, moving on, we're going to go look at the genomic context section. This tells you the approximate position on the chromosome 17q21, while the sequence information tells you that this gene goes from 43,044,295 to 43,125,483, and the complement means that it's on the reverse strand. If there's no complement displayed, that means it's on the forward of the strand. The position of the gene on the previous assembly, GRCH37.patch13, is also shown. Note that these positions are different. A cartoon of the genomic region with the gene is next displayed, and the arrow for BRCA1 tells you that this is on the reverse strand. And there are some overlapping and nearby genes as well with their strandedness displayed. The genomic regions, transcripts, and products section shows all the different transcripts for this gene, and BRCA1 is a good example because there's been lots of research on this gene. Note that the genomic sequence box will tell you that the coordinates are for GRCH38. The consensus or canonical gene model is shown with the green vertical lines indicating exons. An overlapping gene, NBR2, is also shown. If you select and click on the gene model, it will expand to show many transcripts in blue and corresponding proteins right below in red. So this protein that I'm showing right here is created by this transcript. The transcript or mRNA's IDs start with NM or XM. NM transcripts have been curated while XM transcripts are predicted. For other RNA that doesn't translate to protein, those starting with NR have been reviewed while XR are predicted. Again, we have an NR example here of a misc RNA, and you can see there's no corresponding protein underneath it. The same NP and XP naming scheme applies to proteins as well. I'm going to skip over the bibliography and phenotypes section and go to the variation section and click on Gene Viewer Report, which will take you to a dbSNP tool where you can filter the SNPs on a gene. By default, it displays all the coding SNPs in that gene. So this is a useful tool that you can explore further. Going back to the report page, I will skip over HIV-1 interactions, pathways, interactions, general gene information, and general protein information. Let's explore the NCBI reference sequence section a little bit more. I don't pay any attention to the genomic contig entry. The next few items show reviewed transcripts where there is good confidence in the transcript. The next part of the reference sequence section is the RefSeqs of annotated genomes. And here we say that again, this shows what genomic assembly the coordinates of the gene refer to. It's referenced GRCH38. And then there's a bunch of predicted XM transcripts shown here. And you see that they produce, again, XP proteins. I usually don't pay any attention to the alternate section. And then the suppressed reference sequence shows some old RefSeq, some transcript IDs that have been taken out because they were shown not to have a good confidence. Next is the additional links section, which gives you more places to go and look at information on BRCA1. Quickly, let's visit the right side of the page starting at the top. The table of contents have links to each of the sections of the report we were just looking at. Related information gives you links to information in other sites for BRCA1, should take you right to that actual entry in their database. The links to other resources, which will give you additional links to BRCA1 in other sites. We click on the HGNC link and we'll go right to the HGNC page for BRCA1. The rest of the information more general.